Christophe Espírito Santo. He is from the Agrifood Technological Center of Castelo Branco, CATAA, it's the name of the entity, and I will pass to him to do this presentation. Hope you enjoy, and we will wait for your questions in the end of the presentation. Thank you very much.
Good morning, everyone. Thank you for the nice presentation, and I would like first also to thank the invitation from Cluster to give this talk, and also to you all for listening to this presentation. I will be presenting to you the areas that we are trying to develop in our center. Just to keep you in focus, our center is just recently started applied research on packaging with collaboration with universities and also enterprises. So the areas that we are, I'm trying to change the, oh, okay. Okay. So the areas that we are interested in developing right now are as follows. We are trying to do some intelligent packaging using temperature integrators. Also, we are looking at active packaging. So, for example, developing some edible films using microbiota to protect fruits and vegetables. We also are doing some controlled atmosphere. With this, we also are applying it to modified atmosphere packaging. And I will also talk to you a bit of future developments that we want to look at. Also for the packaging, we are testing some new technologies, and we see an opportunity for the packaging industry to develop in these technologies. So why are we interested in packaging? The main reason is food waste. The longer we keep a product freshness, the longer it can be consumed. 925 million people suffer from malnutrition worldwide. So this is a major problem for the world. 1.3 billion of tons are wasted every year, and this will rise in costs over $750 billion every year. Furthermore, we also throw all the energy that we consume for growing, raising, refrigerate, ship, and also pack food. The environmental cost is very high. With climate change, this will even get worse. So we need to have a more sustainable approach. Our objective is to extend the product lifespan, so keep its freshness with a more sustainable approach, minimizing the environmental and economical costs, using natural ingredients or byproducts, and remove or minimize the addition of chemicals that can lead to health impacts. So this is the main objective that we want for our projects in the packaging field. So let's start with the time temperature integrators. I will be giving you a bit of an overview of these devices. So what are these devices? These devices, we basically are interested in them because the sell-by dates are not really accurate for product freshness. So these devices are basically an alternative to conventional temperature control systems and allow to determine the impact of temperature and/or time on food products. A thermally unstable substance by a temperature increase can undergo a change of shape or color and this change can be quantified. So this is how it works, these devices. So with the temperature change, we can see if the product was properly stored or not. So the main feature of these devices, they are small and resistant. So there are 4 types of temperature integrators: enzymatic, microbiological, chemical, and physical. So the first one is the enzymatic. As the name states, it uses an enzyme to quantify thermal impacts. The accumulation of a reaction product of the enzymatic catalysis shows how serious is the impact. As you can observe in the graph, at low temperatures, the enzyme activity is low. So low amounts of products are formed. As the temperature increases, more and more product is produced, and the enzyme until the enzyme reaches the optimum temperature. After a certain temperature, the enzyme denaturates, and there is total loss of activity. So as you can see in the graph, we can see that at low temperatures, the enzyme doesn't work as fast. So we can basically monitor if the product is properly stored. As the temperature increases, then there's the enzyme gets faster in the activity, and so there's more product being released, so we can see a change in the color or shape, and this is indicative of a thermal impact. So we can have multiple ways to build a temperature integrator. We can have an enzyme that can make sure the product does not reach a certain temperature, and only after a very long period of in storage temperature, all the substrate is converted into a product. Meaning that even at correct storage conditions, the product loses its freshness with time. If a thermal impact occurs, the reaction will be faster, as I said before. Another way to apply these enzymes is to use thermally stable enzymes for correct sterility or pasteurization cycles. So as shown in the graph, the enzymes denaturate after being exposed to a period of time at a certain temperature. By checking if there's any residual activity, we can make sure if the cycles were performed correctly or not. The advantage is that we can modulate and calibrate the temperature range of these integrators as we need, and by the food products that we are using. So here we have an example of an enzymatic time temperature integrator, which is the Vita SAP. This is a simple adhesive sticker that is put on top of the food product. And it's a simple enzymatic system with a color change caused by pH decrease. The capsule, as you can see, is when it's not activated as no color. When it's activated, it changes to green. And as time passes or temperature increases, there's a lipase enzyme that does hydrolysis of a lipid substrate, which causes acid release and pH decreases. So this will in turn change color from green to bright yellow and then to red. Combining several types of these enzymes with different concentrations provides a variety of responses depending on the temperature. This indicator, as I said before, provides information at all temperatures. At certain temperatures, these devices have low activity, so the color change is slower. At higher temperatures, the color increases following the enzymatic activity, and it's irreversible. As shown in the image, initially the label is colorless, and then with time and temperature, the color changes to to orange and red, indicating the loss of product quality and freshness. The advantage of these devices, as I said before, is they are small, low cost, they are wireless, so they are shock resistant, and they are really easy to use and also to be monitored. So a consumer can easily identify if the product is fresh or not. The next type of time temperature integrator are the microbial. These are the mostly used devices. They can be divided in two types. The first type that I'm showing you is the process impact that's based on the quantification of the number of surviving microorganisms. After a sterilization cycle, a thermal-resistant microbe can be inactivated if the cycle is done correctly, or if it grows, then the cycle was not properly done. Microbes used here are normally spores of microbes that resist to very high temperatures, thermophiles, isolated from extreme environments with high temperatures such as close to volcanoes or deep sea hydrothermal vents. These microbes evolved to have thermal resistant enzymes that keep on working even after being exposed to temperature over 100 degrees. Checking sterilization cycles is very important because spores are very hard to inactivate and can germinate after an incorrect cycle. For example, we use spores of Bacillus terrathiomorphulus as a biological indicator for sterilization processes using steam. The second type of microbial time temperature integrator detects if there are or not microbial growth. So as the figure shows, increasing the temperature leads to increase of microbial proliferation. It's more or less similar as you can see with enzymatic activity. This feature is used to measure if a food product was properly stored at correct temperature. At low temperatures, the microbes has a very low proliferation rate, meaning few new cells are being created. If there is a temperature increase, the rate increases, leading to high amount of cells. This can be monitored visually, and the consumer can also detect really easily if there's an improper storage condition. The microorganisms used can be chosen according to the food product and the temperature range that we need. Here I show you an example of this microbiological time temperature integrator, which consists on a pH variation. So the color gradually changes from green to red by a pH decrease. So depending on the application, this device can be adjusted by selecting the right microorganisms and gel composition. So as the microbe grows, more acid is being produced and the pH is being decreased. So these labels are normally stored at minus 20 degrees or minus 18 degrees, so to prevent bacterial growth. They are really easy to be applied in the package just by thawing it at room temperature before applying it, and they are applied to the food package. And when there is a temperature increase, then the microbes grow and cause the pH to decrease, and then there's a irreversible change in the color. So the next device is the chemical time temperature integrators. As the name says, it uses a chemical. The chemical is photosensitive and it changes the color with time and temperature. So this device is very simple to apply and to read. The end consumer can get information very easily. So as you can see in the image, the label can be stored at room temperature, but protected from light. Then it's activated by ultraviolet radiation, so it becomes blue or another color. Then it's covered with a UV protection layer, and then with time or temperature, the color basically fades away. The molecule needs to revert to colorless based on a air-universe equation. These labels can be stored, as I said, at room temperature, but they need to be protected from light. If not, then the photochemical will be degraded. So, as an example, we can have the Cool View and also the Best Buy. These are all activated by UV radiation. As you can see on the right, we can see that at day 0, when it's activated, it becomes blue, and then with time, the color fades away when it's stored at 1 degree. And if it's improperly stored at 25, for example, at 25 degrees, then the color change is faster, so it becomes colorless faster. The enterprise has many different kinds of applications for this, so we can find different labels that basically work the same way and tells us if the product is fresh or not, and also it's adapted to different temperature ranges. The next one, we have a physical time temperature integrator. This integrator works on a physical factor, diffusion. Under a certain temperature, color chemical basically melts and travels across absorbent paper. As the temperature increases, more compounds melts and further it travels in the absorbent paper. So this is a very simple device that helps us know how much time the product was in a certain temperature. As an example, we have the 3M Monitor Mark. There are many models with different temperature ranges, but they all have the same principle. As I said, if they are exposed at a certain temperature, the compound melts and then it travels further in the absorbent paper. So the time temperature integrators have many advantages. They are very simple, inexpensive, small, resistant, very intuitive to work, and consumers can easily identify product freshness. They also can be applied in different packages, and they can be calibrated in different temperatures. So these devices we are also developing in collaboration with our other universities so we can avoid to waste products that are still fresh and they probably are not in the same they are not in the correct sell-by date. So if we properly store a product, we can extend the lifespan of this product. With this, we change also a bit of the topic. We are also interested in the active packaging. So the active packaging is the incorporation of certain components into packaging systems that release or absorb substances from or into the packaged foods or surrounding environment, so as to prolong the shelf life and sustain the quality, safety, and sensory characteristics of the food. Our main focus is carbon dioxide and oxygen and moisture for the modified atmosphere packaging, which I will talk about later on. and also the ethylene scavenging. We are particularly interested in ethylene because most of our work done in modified atmosphere packaging is for fruits. Um, so the active releases systems, we are also very interested in antimicrobial agents and antioxidant agents, in particular with essential oils. We are evaluating the antimicrobial and antibiofilm activity of some essential oils, and hopefully in next year we will start implementing these essential oils into packages. We are also looking at metals that can be added to the package layer, such as silver or copper nanoparticles. The only problem with this component is that it's a metal, so it can lead to some contaminations, and this kind of work is a bit different than using a natural product, such as the essential oils. In this part of the active packaging, we also doing work in edible films. So we are basically isolating microbes from fruits or vegetables. We select them, and then we coat products with this microbiota and see which one is protecting the fruits. And then with this, we can develop edible film with different materials. And also, now I will talk to you about the controlled atmosphere that we are developing now. It's a technology that some local producers are looking into to use and also to benefit from. Basically, it consists in modulating the percentage of three main air gases. In this case, for fruits, the objective is always to lower the oxygen. For products such as meat, O2 levels need to be higher, so we can keep the meat bright red, because consumers prefer bright red color. Just because oxygen reacts with myoglobin and keeps this red coloration. So for fruits, we try to lower the oxygen and increase the CO2 levels. I will show you how we do this in a chamber. So the control atmospheres for fruits and vegetables, we reduce the oxygen, increasing the carbon dioxide, and remove the ethylene. For the atmosphere is constantly renewed by nitrogen injection using a generator, and also the CO2 is also injected by a generator. The oxygen is basically just controlled by opening letting air in the chamber. So we have a computer that controls the amounts of these gases, and so we can control different atmospheres and do some lifespan studies using different fruits. So the benefits from control atmospheres, it's basically delayed maturation. We can also reduce the rate of respiration. We also reduce the production of ethylene, so we have a reduction in ripening, and also we can control. We also have insect control. The main disadvantage is that we can happen that we have irregular ripening. This kind of, if we choose the incorrect atmosphere, it can lead to off flavors and also some taints. This also can lead to physiological defects or rotting after fruits being removed from the atmosphere. So this is accelerated. This technology is not that recent. The studies from the 1940s have characterized most of the conditions for fruits and vegetables to be conserved at different temperatures and atmospheres. And now we can also have big containers that are shipped across the oceans with fruits and vegetables using controlled atmospheres, as you can see on the top images. In our facilities, we have refrigerated chambers, which that each has 4 pallets with different atmospheres in each one, so we can perform our tests. For now, we have successfully completed a test with cherry fruits, and we are now in the second month of the peach conservation using different atmospheres, and it looks to be promising. It's a technology that local producers don't use, and they need to centers like ours to show them how it's done and how they can apply to their conservation techniques. So now let's go back to packaging. This technology can be applied to closed packages. So, but however, it's very different than controlled atmospheres. Controlled atmospheres have constant renewal of gases. Modified atmosphere packaging does not have a monitoring system to control the amount of gases and generators to balance the amount of gases. The fruits inserted in the package are sealed with different atmospheres. The film needs to be permeable to allow some oxygen to enter, and because we cannot have low oxygen, if not, it will lead to undesired anaerobic fermentations, which can lead to off flavors. And the excess carbon dioxide also should be removed, because high carbon dioxide can also lead to changes in taste. And also, let's not forget about ethylene. This hormone needs to be trapped, if not, the fruit will ripen very quickly. Okay, so just to summarize, the high carbon dioxide helps to inhibit microbes, but also it can lead to changes in taste. The high amounts of oxygen can lead to oxidation, and so quality loss, and also microbial growth. Low levels of oxygen prevents oxidation and rancification. It also prevents growth of aerobic bacteria, and it slows down the metabolic rate of food. Ethylene, when it's present, it accelerates ripening. The moisture, if it's high levels, it can promote fungal growth, and if it's very low, also food products will lose a lot of water, so changing the quality. Comparing the two techniques, to the modified atmosphere packaging and controlled atmospheres. So one, we have in the modified atmosphere packaging, we have uncontrolled amount of gases. I say uncontrolled because we don't have a monitoring system, but if packaging systems evolve or technologies evolve, we probably can control the amount of gases at different temperatures. Using controlled atmospheres, we can control and monitor the amount of the gases. In using the modified atmosphere packaging, the atmosphere is not renewed. On the other way, using controlled atmospheres, it's always renewed. The modified atmosphere packaging is practical, and it can reach final consumers. So it's very practical for the point of view of the consumer. For the controlled atmospheres, it's not practical. It depends on bulky equipment and expensive equipment, and it only reaches retails. It cannot reach the consumer. So the only retails will get the fruits or vegetables that are conserved in the controlled atmosphere. So the challenge of this kind of products is to develop, or this kind of products using modified atmosphere packaging, is to develop films that can modulate oxygen, carbon dioxide, and moisture, and trap ethylene at different temperatures, because the 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 food product or the fruit has a respiration rate. When there's a temperature change, then the respiration rate is different. So more carbon dioxide is produced and more oxygen is being consumed. So the package needs to respond at different temperatures to the temperature difference. So the package needs to adapt very quickly to a temperature change. If not, the fruit will risk to be wasted very quickly. So the future developments that we are looking into this field is a mixture of both technologies using modified atmosphere packaging and time temperature integrators. We believe that using these two technologies will get the products with longer lifespan. And we also are looking into development of films that exchange oxygen and carbon dioxide at different temperatures, and always balance oxygen and carbon dioxide, independent of the respiration rate of the fruit or vegetable. So let's not forget the environment. So the use of biodegradable materials might be the answer to control oxygen and carbon dioxide. So this is a field that we are also looking into it. Let's not forget that Portugal is one of the biggest producers of cork, which is a natural material, and it's used for bottling and aging of wines worldwide and for many centuries. So this is an example for us to keep motivated to find something that is biodegradable and that can help monitor, or not monitor, but control oxygen levels and carbon dioxide levels. So on another topic that we are recently being testing is pascalization or high hydrostatic pressure to different food products. So this technology uses high pressure to basically eliminate microbes from the product. So how does it work? The food is inserted into a pressure vessel. The water is filled at low pressure. Then when the vessel is full, very high pressure is applied. Just to give you an idea, it goes to 200 millipascal to 800 millipascal. This causes damage into the cell membranes of microorganisms and also denaturate its proteins. After a certain time and pressure, the product is removed from the vessel. This technique is very recent and is very appealing for the food production industry. This treatment inactivates microbes, generally leading to longer lifespan of the products, and also it reduces the addition of preservatives. Meat producers, for example, look at this technique to eliminate the addition of nitrates. So why use this technology? As I said, it inactivates all microorganisms. Also, it can inactivate spores and enzymes. There's a minimal impact on quality and nutrition. It does not depend on mass or size of the product, and it has the possibility to reduce the amount of preservatives. So why I'm talking about this technique on a packaging presentation is just because the packages used for this technology are a certain number of plastics. We cannot use glass materials or other materials. So I think after this technology being really established in the food industry, we need to rethink packages to be used in this technology. We need to have a more sustainable approach, try to avoid non-biodegradable products. So with this, I would like to talk about biodegradable packaging. With the food waste, it also comes higher amounts of packaging waste. Most materials used for food packaging are not biodegradable or compostable. Therefore, this represents a major impact on the environment. Food packaging stability is an advantage during shelf life of the product. However, it turns to a disadvantage when packages are no longer in use. So new strategies are needed to reduce the waste across the whole supply chain. As observed in the Eurostat graph, we are still very far away from reusing all the waste we produce. We still see a large gap between the waste generated and the waste that is recovered or recycled. Agri-food byproducts can help in building new materials that can be used as package for different usage, maximizing agri-food industry opportunities and minimizing the environmental impact. The development of new materials that include biopolymers for food packaging represent an area of key development nowadays. These materials can help to address the problem of waste that we have nowadays, since they are eco-friendly and biodegradable. There are 4 types of biodegradable biopolymers according to their synthetic route. One, those obtained from biomass. The second, obtained from microbial production. Third, from chemically synthesized from agro resources. And also obtained from chemical synthesis from fossil resources. So new techniques, it is, however, important to meet the food packaging requirements for this kind of packaging. Therefore, new technologies are being developed to ameliorate properties of biopolymer matrices for food packaging, for instance, to reduce or prevent acidity, color change or loss, nutritional loss, or dehydration, microbial proliferation. Addition of natural additives, as I talked before, is essential to meet this requirement. With this, I would like to thank you for your attention, and I'm open for questions.
As there are no other questions, it seems like there are no other questions. I would like to thank you, Christoph, first of all, and I would like to ask you, could you maybe go to the slide where you had the TTI, and where you had the different colors from green to orange, red. It was very at the beginning. Or actually, I can go there as I can. Okay, this one. Um, I have a question because, for example, I know that Alexander is having problems to see red and green. So for me, the question is it really with this kind of system? Because as I can see, 25, it's a green for me, 40 is a green for me as well, 65. I wouldn't be sure if this is already a orange or if this is already a red. No, it's not a red, but it seems like kind of orange. So I'm not quite sure if I would throw away this earlier as it is actually, because it's not, for I think for consumers it's easier when you have thumbs up or thumb down kind of
Yes.
example, saying okay, everything is fine, because this would confuses me, and when I think about people who can't see red and green, it will be difficult. For older people, could be difficult as well. And I'm not quite sure if we have the best before problem solved with this kind of temperature.
Yes, you raised an interesting question. This is something that our, our integrators that we want to develop, we want to be more intuitive and more easy. We don't need to have such color change because it's very hard to see. And we didn't address the problem of colorblind people that probably cannot see this kind of uh, color changes. We need to have a more simple system. As you can see in the picture, the green box is still fresh product. So from 25 to 85% it's still fresh. Over 100% then it's not fresh. Using this kind of uh, enzymatic temperature integrator. Um, I personally and our collaborators also prefer to have a simpler device that can basically or fade away the like a sentence saying product fresh and then it's removed over time, or also appearing as some sentence saying the product is no longer fresh. Something that is more visually easy to identify. Or also, we are also looking if we want to have a scale, then we need to have like attributes like a number instead of a color. So some systems already use this. For example, I can show you the chemical TTI from Coolview and Best Buy. They have also a number system that shows if like a temperature history of the product. It's more used for storage or like more for retail, so they can know if the product was frozen, then defrosted, or and then frozen again, or different kinds. So it attributes a number, and then the number we can look at a table, and so we can see what happened to the product. I hope I answered your question.
Yeah, totally. And actually I don't know how many people are really colorblind, so that's actually not the biggest issue, I think. And I totally understand green always means good and whatever, and red
Yes.
is not good. So I can totally understand the choose of the color. And what I was discussing with my colleague here as well, that it would be great if it could be, one icon which is used for all. I know this depends always on the company, but it would be great if it would be just one icon which can be used and which is then, used to it. And it's not because we were discussing about this during your presentation, how it is with just names or just it's good or it's not good, but this is always then related to the language. So
Yes.
if you have a company, of course they have to produce this for different in different languages. So I think if it would be just an easy icon, I think that would be the
Yes.
easiest solution. And even with the system here, with the blue heart, apple, whatever it is, that's for me the easiest thing would be blue or white. That's it. No, nothing in between.
Yes.
Because otherwise I will be confused when I see, for example, I don't know, 3 days at 15 degrees. I wouldn't be sure if I still can eat it or not.
Yes, that's also it needs to be uniformized the system because different companies have different scales, different colors, different. So this can be confusing
But
for.
why do they have scales? This is my actually my real question. Why do they have scales? Because scale for me always
Yes.
does it have to be due to some regulations, or because for me, the easiest way is saying black or white. White is not good, black is good. That's it. I don't have to measure any color or scale or whatever.
Yes, for a consumer point of view, yes, you're right. It's just easier to see if it's good or not. But also, these devices are also used in the industry to see if a certain producer conserved the fruit correctly, or the fruit or the food correctly. So, for example, it can be frozen and then defrosted, and so they need to know, and so then scales make sense.
Yeah, make totally sense. Yeah, yeah. Okay.
But
Okay, this
but it's something
yeah, I
that they
just
need
saw
to
it from the
train.
consumer side. Sorry.
Yeah.
Yeah.
But the main goal, I mean, for avoiding food waste, I think the main goal is like you said, it's something that needs to be simple with the icon that can be universal.
Yeah, right. That would, at least for consumer, would be the easiest
Yes.
solution, I think. Okay, thank you. Are there any other questions from the participants? Doesn't look like. Susanna, would you like to say anything?
Susanna is because we have one microphone, and so Susanna
Okay.
didn't hear you, but Katrina asked
Yeah, you have the
you
microphone. Okay, okay.
if you want to say something.
So, okay.
She wants to say something. I will pass Susanna.
Okay.
Hello?
Hello, we can hear you.
Yeah, I don't know if you heard me just after Christophe finished his presentation.
No, we didn't.
No, okay. I would like to, if there is no answers from you anymore, I would just like to thank you all for attending the webinar. And I would like to say that I was very impressed with the amount of work done by, or that is being done by CATAA in these packaging fields or areas. Christophe is the scientific director of CATAA, and although we are working in the same building, we actually we don't really know what each one of us is doing. So it was a surprise for me. I think that we understood that Also, there are many difficulties that CATAA has encountered in this work that he's been doing. And I think I would like to understand them as challenges and that can be solved with the cooperation between entities and that the AdPack project can provide. Actually, there are lots of things that Christophe was talking about that could be challenges, like the packaging materials for the high pressurized techniques that are being used now for storage of food. I think that this project can help CATAA and all other entities to build cooperation projects to solve common problems and actually develop new solutions for these packaging areas. So I think that I can say that he is also available. I think that his email is in the presentation. If not, we can provide it to who needs it, and we are available for all kind of cooperation and doubts that you might have.
Thank you very much, Susanna. And of course, again, thank you very much, Christophe. It was really a very, very interesting presentation. I think all of us learned a lot today what you are doing and that you're really into intelligent packaging. I would also like to thank thank you to all the participants, and I would like to remind you to our next webinar, which will be on the 8th of November at 11 for everybody who's not from Portugal, for all the Portuguese people at 10 a.m. And the topic will be packaging and nanotechnology and plasma technology. And yeah, I would like to welcome you to this webinar as well. And yeah, if you have any questions or if you would like to have the email address of Christophe, just send us an email and we will come back to you. And then I wish you a very nice afternoon. And thank you so much.